Welcome back to another episode of CSI, The Science Revealed.
Today we're going to be revealing the science behind muscle contractions and rigor mortis.
Here we have a neuromuscular junction model. In the purple we have a motor neuron, and in the blue and green we have a muscle fiber. It all begins as an action potential travels down the axon of a motor neuron. The action potential reaches the axon terminal, where it triggers vesicles filled with neurotransmitters to move towards the border of the axon terminal where it meets the synapse. Neurotransmitters will leave the vesicle and enter the synapse and travel to receptor sites that are on the sarcolemma. The sarcolemma is a specialized membrane surrounding the muscle fiber. The receptor sites will trigger the sodium and potassium pump to regulate the sodium and potassium entering and leaving the cell. 3 sodium will leave the muscle and 1 potassium will enter the muscle. This creates depolarization, which further creates an action potential that will travel across the sarcolemma. As the action potential travels across the sarcolemma, the outside will be negative and the inside will be positive. The action potential travels down the T-tubule and further hits a DHP receptor. This DHP receptor then carries the action potential across the sarcoplasm and to the sarcoplasmic reticulum.
So after the action potential triggers the sarcoplasmic reticulum, which is a membrane that regulates the calcium ion concentrations, calcium ions are released across the membrane through active transport into the cytoplasm of the muscle cell, also called the sarcoplasm. This requires ATP. The calcium then binds to troponin, which is represented by this yellow beads. Troponin is a complex of 3 proteins attached to the tropomyosin. Tropomyosin will then move to reveal binding sites and will allow for the actin-myosin interaction. The binding sites are these black dashes on the actin. Myosin heads will then use ATP to go into high energy configuration. It will take away one of the phosphate groups to make ADP and pi. This energy allows the myosin heads to bind to the actin, forming a cross bridge.
So after the cross bridge is formed, the ADP and phosphate group are released, and the myosin head moves to its low energy configuration, and it remains in the cross bridge. And this does not require ATP because it's moving from high to low energy, and this is called the power stroke. So the actins pull towards the M line, and it's pulled towards the center of the sarcomere, so that makes the sarcomere smaller. Then the myosin head is still attached to the actin, so then this is called rigor, and there's more ATP that comes in to break it, and then the process starts again post-rigor.
After death, there's no more ATP for the calcium ions to be regulated. So the sarcoplasmic reticulum will let out too much calcium ions that will then go to the troponin, moving the tropomyosin, and it will move the tropomyosin to reveal the active sites. There is enough ATP left to keep the myosin in high energy configuration.
So since all of the myosin heads are in high energy configuration, the muscle contracts because that doesn't take any ATP, but then there's no ATP left to break the cross bridge, so then it remains contracted, and then after roughly 72 hours the bacteria breaks down the myosin head, so then the muscle relaxes.
Thank you for watching CSI, The Science Revealed. We hope you enjoyed our film.